So I'm going to ask, can we take one question? Yes. Good afternoon.
Yes. Good. So I'm Bruce Kennedy. I'm a member of the Round Table, and I'm also from Chapman University in California. And my charge this afternoon is to represent those questions that have come in from the public that have been viewing the webinar. And we did get one question this morning. It seems timely for what we were just discussing now. So I'll read you what Barbara Hansen from University of South Florida says. Notably high use of word animal to refer almost always to mice. There have been more than 400 cures for type 1 diabetes in mice and none in humans. It seems that there should be a great increase in the use of non-human primates, adults, not babies, infants, in these sorts of assessments, in development of excellence in quality control, and especially in study designs. Consider this further. And as I was listening to you, and the comment that Daryl said about research is critical and the patients are going to be expecting that, I thought this was timely to bring out. It is a good thing to discuss, because I also feel that we're going to have an increase in animal activism of some sort. That would be the phrase I'd use for now. So, please respond.
Would you start? You want to start? Well, I do. It's a difficult question to engage around. I think that in some ways we walk before we run, and I think building the case to the public that animal models of more comfortable sorts are essential for the conduct of precision medicine research is the starting place, starting with mice models, rodent models, etc., building up public support for that before engaging in conversations about the use of non-human primates in research is likely to be a more effective strategy. If we jump into the deep end, to use that analogy, and try to engage members of the public on the need for non-human primate models for the purpose of precision medicine research, I think we're far more likely to generate a more intense form of political activism in response. So that would be my initial thought about that.
And I'll just add that there's a separation, and I think Richard really brought it to his presentation well, this need for more than just community discussion. There's lots of different platforms to have this discussion, but we're going to see a difference in opinion between real patient advocacy groups or patients versus the public. And especially when you get to advanced animal models like primates and even in dogs, when you use them, the public who do not have a particular disease or family member with a disease will have probably a very different perspective than those patients and the family members of those patients that do, and they'll see a different priority for those types of research.
I'd like to follow up from another perspective. I teach veterinary technicians about laboratory animal science. It's a 10-week course for 30 hours. They come into my class initially, and I think many of them carrying their PETA cards, and they're not so sure about what Professor Kennedy is going to tell them in the course of the course. By the end of the class, we have talked about companion animals and dogs and so forth, and that's a very good way for me to get them to, I don't want to say persuade them, but at least for them to understand what they're getting into as veterinary technicians when Dr. Brown pulls that medicine off the shelf and it's for Fido or it's for the kitty or something like that. So companion animal is a great way for me to convey the message to my students.
Well, and I'll follow up on that just to say that veterinarians have been using human drugs forever, right? And veterinarians do have drugs that were designed and developed and specifically marketed and licensed, et cetera, for animals. But veterinarians actually serve a really critical role here because we actually sit at a nice nexus between all of these species and also in human health. We've mirrored the vast majority of our training programs off the human medical training platforms. And I think that naturally occurring disease models, I mean, obviously I can speak specifically from the cancer space, but I mean, dogs get diabetes, dogs go into heart failure. There are lots of autoimmune and degenerative diseases and all kinds of things that we could be thinking about and taking into consideration that can really complement, and I don't want to say as far as that we could reduce the use of other species. I don't think that it's right for me to say that, but that's a nice dream for us to reduce, you know, all animal use, but really think very critically about what we can learn and learn as much as we can. And veterinary medicine, when we talk to clients, like when I used to recruit for the clinical trials that I now run, a clinical trial is offered alongside a spectrum of other treatment choices. So pet owners get to decide what they want to do. They can, so in the case of a diabetes situation, they could say, yes, I will treat my dog for diabetes with the standard regime that is described in the veterinary textbooks, or I could be offered a clinical trial of a new diabetes/dietary regimen that maybe has some funding behind it, maybe it doesn't, but they have that choice to contribute to the scientific questions at hand. Or they can simply say, I don't want to treat my dog for this disease. And so the pet owner has the ultimate flexibility there. And I think it's a privilege for us to be able to contribute to that space and to really offer the opportunity to give back to science, but the pet owner doesn't feel obligated to do that in any way.
But it's been interesting, I think, in the Parkinson's space, we obviously use a whole variety of different model systems, including non-human primates, but I think in the case of non-human primates, it's been more in the context of models of Parkinson's symptoms, because you can sort of knock out the dopamine system in a non-human primate and get, again, features that are sort of somewhat resemblant of the human condition. People have tried to use non-human primates for sort of more disease-modifying approaches, but largely I think that hasn't been very predictive of the human condition. So in many ways, a lot of the concepts for modeling in Parkinson's have focused on the idea of using more rodent models, to be honest, as mechanisms that you're testing against, and as opposed to Parkinson's disease per se. So if you're interested in protein accumulation or synuclein pathology spread or something like that, that's really what you're trying to model in a rodent system, and then therefore you're targeting your drug against that. And you may never then need to even go to a non-human primate before you can start the clinical trials. And so I think there's, again, that doesn't solve the problem of use of animal research in Parkinson's disease, but at least I think provides sort of a refined understanding of making sure you're asking the right question in the right model and then using sort of thoughtful guidance in that.
So that'll end our panel, and I think we're going to have some closing comments from our co-chair.